The next generation of CTC technology being developed by Janssen in collaboration with Massachusetts General Hospital has been somewhat published and is currently referred to as the iChip, and we're very excited about this technology. It will build very much on what cell search has done, so we don't look to necessarily replace the enumeration and the types of cells that are currently analyzed by cell search, but we hope to expand that. We're going to get some different kinds of cells and improve and expand the ability to look at these cells phenotypically and genotypically. The cell search technology is based upon a positive selection of cells. In other words, the cells, the circulating tumor cells in blood, if they express EpCAM, or epithelial cell adhesion molecule, are captured by the cell search technology, and those are the cells we enumerate and count. And we can do some molecular and phenotypic expression, as I explained in the clinical trials. Nevertheless, we know that this is just the first generation, and there's a wealth of additional information that can be gleaned from those cells based upon their molecular expression, based upon additional phenotypic analysis of those cells. We also know that there are circulating tumor cells that do not express EpCAM. We don't know how many for sure, but we know that there are some there that do, and these may be very important in the progression of the disease and metastatic spread. So the iChip does not require the cell to express the EpCAM molecule, so we'll be able to obtain cells that are both EpCAM positive as well as EpCAM negative, and then examine these in a variety of ways, both phenotypically and by expanded genotypic analysis. The need for liquid biopsies in solid tumors comes from the fact that when two different aspects of metastatic spread. First, many times there's more than one metastasis, and we can't assume that each metastasis is genetically identical. In fact, we know that each metastasis is perhaps a little bit different than the other metastases. So if one takes a biopsy of a metastasis, you can get sort of a molecular image of what's going on in the tumor, but it's only a partial image. And we believe liquid biopsies are a more representative sampling of the entire tumor burden in that patient at that time. Plus, the cells circulating in the blood are highly correlated with progression-free survival and overall survival. So it appears that the cells in the blood are particularly involved in the whole process of metastasis and spread. So they may be the most important cells to really understand what those cells are doing at a particular time. Metastatic or liquid biopsy can help us by getting a better representation of the cells in that particular patient. It can also help avoid the pain, discomfort, and complications of metastatic biopsies. Sometimes biopsies, and particularly in lung cancer, it's sometimes very difficult to even get a biopsy, and it can provide a means of getting tumor material in a relatively non-invasive way. This then opens up the door to more frequent and easier and less costly ways of monitoring the patient in real time, a way of getting ahead of the tumor rather than wait to see whether or not we've provided the right therapy, but actually provide a therapy, monitor how it's doing, see if the cancer is adapting or changing in response to that therapy, and then actually provide the next targeted therapy before that portion of that subset has a chance to expand. Well, clearly, for tools within the that Janssen is currently developing in the area of this liquid biopsy is the whole, this the thrust behind the whole cell search, as well as our new next generation iChip technology. It's really a combination between Janssen and the researchers at Massachusetts General Hospital, where we're really bringing together both rather sophisticated engineering expertise, medical expertise, and long expertise in circulating tumor cells in order to really enable in a very open way, in a very rapid way, to get at these circulating tumor cells and all the different populations of circulating tumor cells, so we can better monitor and more effectively create a personalized way of analyzing what your particular cancer is doing right now and in response to the therapy that you're on.